Alright, so this will be on DNA replication, 13.2 and 13.3. So DNA replication is what happens in S phase of mitosis and meiosis. So duplicating or doubling our DNA, we need to do that to make new genetically identical cells and to make gametes. And it's not a long process. There's just a little bit of complications in that there's a lot of different actors, a lot of different players in this process. And a lot of them we won't have to learn about, but some of them we will. Some of them will be important, so I want to make sure that we pay attention to who those are. So an overview, what's going on? What's going on is we have one parent strand of DNA, and we make two daughter strands. And we do that by first unwinding our DNA so that it's two kind of single strands that are separated from each other. And then we add the complementary base pairs to each strand, nucleotide by nucleotide. So as you see these arrows moving up and moving down, adding nucleotide by nucleotide, synthesizing a new strand of DNA. And then when we reach the end, we have two new strands of DNA. Each strand has one strand, each new double-stranded piece of DNA has one strand that came from the parent strand and one brand new strand that was synthesized nucleotide by nucleotide from the template strand. This is what we call semi-conservative because it only synthesizes one brand new strand per new double helix of DNA and utilizes one parent strand. So half is old, half is new. Alright, before we get started with the specifics of DNA replication, after today, this is things that you should be able to know. You should know the meaning and function of these words. The AP test expects it. Anything additional than these words or these concepts that were in this chapter may be beneficial, but not required. The enzymes that you need to know what they do that are involved in DNA replication are polymerase, DNA polymerase, 2, RNA primase, 3, DNA ligase, 4, helicase, and 5, topoisomerase. Those are it. There's hundreds of enzymes and different molecules involved. 5 are the ones that you need to know. These other things we'll be talking about here in the rest of the video. Make sure that after you watch, you go back, and if you can't answer one of these things, answer it with a yes. Go back to that section of the video. Alright, so we're going to take a closer look at DNA replication now. So first, DNA starts by we initiate DNA replication. We start it. And the first thing we need to do is, one, we need to unwind the DNA first. We need to expose the two strands so we can build in new nucleotides in between. So helicase is an enzyme, and its job, here's a picture of helicase, is to go down and unzip or unwind DNA. Because DNA is a twisted double helix, to be able to unzip or unwind DNA, you kind of have to straighten it out and hold it and to relieve the strain. And that's what this enzyme topoisomerase does. It relieves the strain and kind of holds on to DNA so helicase can unzip it. So we hold it and we unzip. The next thing we need to do, oh, by the way, these single-stranded binding proteins, we don't need to know their function for your information, they kind of steady or hold on to the two single strands and keep them from coming back together. But anyway, now we have it held and we have it unzipped. What we need to do is make start signals. Tell where exactly on this unzipped DNA are we going to start our DNA replication from. The machinery that actually adds new nucleotides can't begin synthesizing just out of thin air. It needs a start signal. And what RNA primase does is it lays down an RNA primer, or start, to tell those enzymes that are now going to add the nucleotides where to start. A few important features to know is this point, this V, where helicase is actively unzipping. That's called the replication fork. It's called the replication fork, or the origin of replication. The replication fork, this V right at the crux of which new DNA is being unzipped. So that's how we start DNA replication. Topoisomerase holds on, helicase unzips, forms a replication fork, primase lays down these start primers to tell where we're going to start adding new DNA nucleotides. Now we're going to make a new DNA strand. Now, important, new DNA can only be created in a 5' to 3' direction. That means when we're making a new strand, this is going to be the 5' end from where we're going, from where we came. The 3' end is going to be towards where we're going. 5' end of the new DNA is where we started, 3' end is where we're going. What that means is that DNA replication will proceed towards the replication fork on one end, and it'll go away from the replication fork on the other end, since the two parent strands are anti-parallel. Their 5' and 3' are mismatched. That means replication will going from 5 to 3 and be in two different directions. The strand, now here's the top strand, where replication moves towards the replication fork. That's called the leading strand. The other one, where the replication goes away from the replication fork, called the lagging strand. Important to know the difference. DNA polymerase, which is this enzyme, is going to be what adds the new nucleotides from a 5' to a 3' direction. So it's taking new nucleotides, a phosphate, ribose sugar, nitrogenous base, and sticking them on here, one at a time. They can proceed in human cells about 50 bases per second, so pretty fast. So on the leading strand, you'll notice it can start here one primer and keep going just uninterrupted as the DNA unzips. However, on the lagging strand, since the DNA polymerase can only go this way, it has to keep going back after more DNA is unzipped by helicase. And that requires a new primer to be laid down and it to fill in the gap between where it left off, and then go back and fill in the gap to where it left off. It needs to keep going back. I mean, it needs to go back and fill in this space, and then as more gets unzipped, go back and fill in this space as more gets unzipped. So here's our replication fork. Here's our lagging strand on the lagging strand, and DNA polymerase going in to fill more in. And it has to go all the way back and start all over again and fill in the empty space. And then a new primer has to be laid down. It has to go all the way back and fill that in. So as you see, leading strand, really simple. Really simple. Lagging strand, not so simple. It's going to take a few other steps in our next slide. So here, where we're completing, where we're ending, termination. Once all DNA has been replicated, the replication machinery, those enzymes, detach, and the DNA winds back up into a double helix. The RNA primers must be removed, though, because the RNA primers are RNA, and they need to be filled in with complementary DNA by DNA polymerase. So, what happens on the leading strand? Pretty much simple. DNA polymerase comes in, the RNA primer leaves, and DNA is filled in the holes. On the lagging strand, there's a whole bunch of these RNA primers. See here? When the DNA polymerase on the lagging strand is synthesizing new DNA and it hits a primer from the last time, it has to go back and start again until it runs into the primer it just laid down. What that leaves is little fragments of DNA interspersed with many primers. These little fragments are called Okazaki fragments, after Dr. Okazaki, a Japanese scientist who discovered them. So, what happens is DNA polymerase goes back and fills in all of these RNA primers, and then a special enzyme called DNA ligase forms bonds between these Okazaki fragments, these little gaps, so that it's all one big DNA molecule. So DNA ligase joins together those fragments of DNA caused by removing the primers and filling them in by DNA. And then we're all done. Then we've got two coiled up new DNA polymers. Right. So here we go in an all-in-one picture. Helicase, unzipping our DNA. Topoisomerase, that's missing right here. Holding on, making sure to relieve the strain. Our RNA primase is adding primers, one at the very beginning of our leading strand, every once in a while on the lagging strand. DNA polymerase on the leading strand synthesizes new DNA one nucleotide at a time towards the replication fork. In a 5', in a 3', or a 5', in a 5' to 3' direction, starting at 5' to 3'. On our lagging strand, RNA polymerase, the 5' end is going to be towards the replication fork, the 3' end is going to be away, so it will have to keep going back, and RNA primase is going to have to keep adding primers. After it's all done, DNA polymerase is going to remove the primers and fill in those primer chunks with DNA, forming little Okazaki fragments, and DNA ligase is going to join those Okazaki fragments together. So DNA replication is very, very, very efficient and very, very accurate. Only one mistake happens in about 10 million base pairs that are replicated. Sometimes mistakes do happen though, or sometimes DNA is damaged by things like UV light. UV light physically damages DNA in your skin cells. That's why skin cancer or melanomas happen, is because ultraviolet light can mutate DNA and can create cancer. So wear your sunscreen, don't go tanning without it. Stay out of the sun, protect yourself, don't mutate your DNA. But it can happen, so your DNA needs a proofreading or a fixing mechanism. That's easy because DNA is complementary. So what happens if this is damaged is it gets cut out by an enzyme called nuclease. DNA polymerase goes and fills it in with the correct DNA, and DNA ligase sutures it or welds it back together. Awesome, awesome, awesome evolutionary development for our DNA to prevent harmful mutations from happening. That's why DNA proofreading evolved. It's a very old, ancient mechanism that's shared by many, many, many creatures. Alright. Last thing I want to talk about is DNA packaging into chromosomes. So DNA is really, really long. Very many, many, many billions of base pairs long. It's so big it can't really fit inside of your cells except that it's coiled up. And it's coiled up around proteins called nucleosomes. Nucleosomes are made of smaller proteins called histones. There's 8 histones into a nucleosome. And the DNA, the double helix, is kind of wrapped around these nucleosomes like beads on a shoestring, or a string around a bead model is what it's called. How closely the DNA is packaged, though, is varied. It can be very tightly wound or very loosely wound. Closely wound DNA is called heterochromatin. Genes on heterochromatin are not synthesized very often because it's so packed together. Protein synthesis machinery can't get in there to read it. It's so packed together, can't get there and read it. When we talk about the control or regulation of DNA synthesis, or protein synthesis rather, packaging or heterochromatin is one way to prevent DNA from being turned into or transcribed and translated into proteins. Euchromatin, on the other hand, is the loosely packaged DNA that's synthesized a lot. You keep needing to synthesize it. So, on our chromosome, some of it's really tightly packaged, like around the centromere. Some of it's more loosely packaged, like out here. And that's because it's very tightly wound or not very tightly wound around these nucleosomes made up of histones. Last and not least, an extension, aging. So, the very ends of DNA chromosomes are called telomeres. What's interesting is that DNA polymerase cannot synthesize all the way to the end of a chromosome. It can't synthesize all the way to the end of the chromosome, which means parts of the telomeres, every single replication, get shorter and shorter. What that means in your cells, every single time one of your cells duplicates, the chromosomes get shorter by a few tens, hundreds of base pairs every single time. What's nice, though, is if that were happening, you'd be, hey, I'm losing genes every single time as I get older. My DNA is not working right. I lose genes that I need. What's nice is the end of these chromosomes, these telomeres, have a repeating pattern, like a T, T, G, 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 T, T, G, 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 or A, C, 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 A, C, 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 C pattern, like this. So like one strand would have T, T, G, 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 and the other one would have an A, C, C, C. And that repeats thousands of times at the end of your chromosome, and your telomeres, and codes for nonsense. That's how your cells protect DNA polymerase from not synthesizing important DNA. However, there's a limit. So, in your gametes, an enzyme called telomerase replenishes or resynthesizes telomeres in your gametes, so that every time you make new gametes, they're not losing DNA that might cause mutations in offspring. But telomerase is not present in somatic or normal body cells, which means over time the telomeres are worn down as the generations go on and on and on. The shortening of telomeres and starting to remove DNA on chromosomes responsible, or is thought to be responsible in part for aging. So a lot of research is going on to how to introduce telomerase and get rid of shortening telomeres in somatic cells to help fight aging. Thought that was pretty cool, wanted to share it with you. See you on Wednesday. All of your telomeres will be shorter by then. Mine will too, but I'll see you then. If you have any questions, let me know.